In this video today, we are going to discuss about gene transcription. Actually, transcription is the first step in gene expression. This process involves copying genes information or DNA sequence to make a RNA molecule. Actually, transcription process run by various kinds of enzymes. Among them, the most important enzyme is RNA polymerase, which actually copy the nucleotides from DNA to RNA and make a RNA template. Now we call it messenger RNA or mRNA. Actually, we know that gene remain in DNA. Actually, in a cell, chromosome or DNA available in supercoil structure. After that, this supercoil using topoisomerase enzyme converted to a plain structure of DNA. Here we can found that this is the supercoil structure of DNA, and using the activity of topoisomerase enzyme, this supercoil structure is converted to plain structure. After that, in the second stage, another enzyme called helicase also act here, and this helicase enzyme actually separate the two strand of DNA and make it suitable place for replication or transcription. After that, this is the plain structure of DNA, two strand of DNA, sense strand and antisense strand. Actually, these two strands are already separated by the RNA helicase, I already told before. And here we can see that a promoter region, this box actually TATA sequence contain a promoter sequence, and this sequence is available before in every gene in the upstream region. And this RNA polymerase is an enzyme actually that bind to this promoter region and initiate translation. Actually, RNA polymerase can run from 5' region to 3' region, so which is complementary to this strand. So this is the promoter sequence, and this green color is also gene sequence. So RNA polymerase first will bind here. We can see the RNA polymerase has complementary sequence to the data box. And this in the next process, after binding in the promoter region, it will start elongation of DNA molecule. And we will found now we will found a complementary sequence of mRNA, which is complementary to DNA. We can see that binding of complementary DNA, complementary mRNA strand. We found a complementary mRNA strand which is already created based on DNA strand, and information already has been copied from DNA to RNA strand. We can see that instead of adding thymine, here added uracil. Uracil base is a characteristics of RNA molecule, because in DNA we can only found adenine, and we do not have thymine in RNA molecule. This is the 5' 3' newly synthesized RNA molecule. Okay. In the next stage, we can see that this molecule, which already created from DNA sequence, this one already created, we call it pre-mRNA, pre-messenger RNA, which is not complete messenger RNA. Here I can describe clearly for understanding pre-mRNA. What is pre-mRNA? Pre-mRNA which contain, actually we know that a gene sequence in DNA that contain also intron and exon. Actually, exons are the sequence of coding genes, that mean that can create a protein and that has information to express. But introns are non-functional sequence which actually not valid. So pre-mRNA contain exon and intron, and after a process we call splicing. Splicing is a process where introns are removed and exons are bind together to complete to construct a functional gene and functional informative mRNA. So that is called mature mRNA. This is the mature mRNA which contain a 5' cap structure, three exon or 3 coding gene combined after splicing, and a polyadenine tail, which is the major characteristics of a mature mRNA. So we can found that a mature mRNA which has been created from a single sequence of DNA. In the last stage, we can see and we can found a complete mature mRNA which is ready to go translation. Actually, this mRNA created inside the nucleus, and this mRNA after the processing will release from the nucleus through nuclear pore and come into the cytoplasm and bind with ribosome to produce protein. In the next video, we'll discuss about translation process, how protein is created. Thank you very much.